One more
Why don't
time.
I start from the beginning?
Please,
Yes,
please
please.
do.
Why don't we start? Okay, I'm going to start from the beginning and then we'll talk. Okay, good. Digestion. Digestive system. Lovely. All right. Let's go back to our friends, the mitochondria, right? What is happening in mitochondria? We're breaking down pyruvate and taking that energy to generate ATP and heat. As a result, we need oxygen for that process, and as a result, we get water and carbon dioxide. Where does the pyruvate come from? We talked about oxygen and carbon dioxide last time when we were talking about respiration, but this time we are talking about the source of pyruvate. And you can get pyruvate from various different sources by breaking down carbohydrates, breaking down fats, breaking down proteins. Usually we are breaking down carbohydrates. Once you run out of carbohydrates, you switch to fats. Once you run out of fats, you switch to breaking down proteins. That is starvation, that's Holocaust when that happens. So mostly it's going to come from carbohydrates. Where do carbohydrates come from? They come from food. They come from the outside world. So we have to eat pretty often because we are warm-blooded animals. We have high metabolism, so we need to eat often. We need to eat a lot. You know, cold-blooded animal like a python, you know, eats a deer and sits there and digests for a month and then hunts again, right? We eat like 3 times a day. So sugars and other molecules are going to come from food. Now, humans are what is called a generalist, and we are also a global species, right? You know, we live everywhere. There are people living in a science station in the Antarctica, on the South Pole. There are people living in Alaska and in Siberia. There are people living in tropical countries. There are people living in submarines. There are people living on the space station, right? And so wherever you live, you're going to try to survive by, getting some stuff to eat. You're going to grab some plants, maybe some fungi, and you're going to grab some animals. And what plants and fungi and animals live is going to be different in different parts of the place. So you eat what there is right around you, or otherwise you starve. And so all those organisms are going to, they're built of proteins and lipids and carbohydrates and nucleic acids, but those are different than ours, right? So if you go out and catch a mammoth, which we often do in this class, we catch mammoths a lot in this class. You catch a mammoth, you are going to eat mammoth protein and mammoth carbohydrates and mammoth lipids and mammoth nucleic acids, and but you don't want those, you want yours. And so what the digestive system does is breaks down all of the mammoth stuff to the smallest possible building blocks. So proteins are broken down to amino acids, individual amino acids. Polysaccharides are broken down to monosaccharides. There are several of them. Nucleic acids are broken down even smaller to the individual A's, individual T's, individual C's, individual G's, individual sugars, either ribose or deoxyribose, and individual phosphates. Complete breakdown. And then you absorb each one of those little elements separately. Lipids, there are 2 types of lipids. There are cholesterol-based lipids, you can take them in, and there are triglycerides, which are broken down to glycerol and the 3 fatty acids. Again, those fatty acids are different from ours. And then what our body does is uses those little Lego pieces to build our own protein, our own nucleic acids, our own lipids, our own sugars. And that means it doesn't really matter what is the source. It can be lab made. It can be GMOs. It can be whatever you got, caught that doesn't have toxins, right? So you don't want to eat, a mushroom that you don't really know because you might be dead in 5 minutes. But as long as you know, because, local lore understands, like, okay, this is edible, this is not. Whatever there is, you can survive because your digestive system is going to break it down to the smallest possible elements, and then your body, your liver, and your muscles and whatnot are going to constitute, build, that's anabolism, the molecules that you need, your own human versions of those. So it really doesn't matter what comes in, as long as you have amino acids, the 20 and essential ones, you have some sugars, you have some fats. There are a few things that you kind of have to take in some small quantities, and those are minerals, and those are some vitamins. Vitamins are kind of a special case, and not every or animal needs all the vitamins. Some animals can produce, for instance, their own vitamin C. We cannot. We have to take it by food. Those are usually small quantities. So you catch that mammoth, and also make some meat with it, and have some fruits and vegetables and salad on the side, and you eat that. And you're putting it into the digestive tract. The digestive tract is a tube that starts at the mouth and ends at the anus. There's no place during that passage through you where there's a big gaping hole. Everything is tightly controlled. All of these little breakdown molecules are going to be actively imported and placed into your body, right? There's going to be some control back and forth about how is it going to go. So, in a sense, officially, formally, the digestive tract is the outside world. It is outside of your body, right? It's outside world passing through you. And everything is very highly controlled, with the epithelia taking or not taking various molecules in and out. There's no hole where the food just dumps in. It's like suddenly it's inside of you. No, it's not. It is the outside world passing through you. You take out of it what you want, and the rest goes out. So digestive system is a long tube, and also has several other organs that are associated with it. For instance, things like teeth and salivary glands, and liver with gallbladder, and pancreas. Those kinds of things are accessory organs that are aiding in digestion. But mostly it's a tube. The food is going through that tube in a kind of a controlled manner. There are several places in the tube where there are gates that are controlling if the food is going to go to the next segment or not, or how much of it is going to go from segment A to segment B, right? So the first one is obvious. Your lips are going to control, are you going to eat or not? Anybody here has a baby who does it, I don't want to eat, right? Yeah, you can do that. So you're controlling, that's your first control. Is anything going to come into your mouth? Then the next one is swallowing, right? Going from the mouth through the pharynx into the esophagus, and that's a very complicated. We actually don't understand all of the neuroscience of swallowing. And I mentioned before, when we were talking about the respiratory system, that the evolution of terrestrial life is the evolution of strategies against choking, because you cannot alter the embryonic patterns, and so the digestive and respiratory tracts cross. They cross in the throat. So the whole process of swallowing is very, very complicated to prevent food going into the respiratory system, which has some smooth muscles, but it's mostly skeletal muscles there, which means you have to consciously decide when it's time to swallow, and then you're swallowing pretty consciously. You can't do it while you're asleep. And so the throat and the very top of the esophagus is going to be skeletal muscle, and then as you're going lower down the esophagus, you lose the skeletal muscle and you gain smooth muscle. And after about a third of the way down, you cannot consciously push that food anymore. It is going to go down by smooth muscles. Remember the Frank Starling law? More you stretch the muscle, stronger the contraction. So if you have a big bolus coming down the esophagus, it's going to stretch the esophagus, and the esophagus is going to push it harder and push it down, 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 until it passes through the cardiac sphincter. Cardiac sphincter is pretty weak. It's a circular muscle, but it's not very good at preventing the bolus from going into the stomach. It's going to go there. If it's liquid, if it's drinking, there's nothing it can do. It cannot completely close. Yes?
Can it also be called the esophageal sphincter?
Yes, it has gastroesophageal sphincter. Cardiac is quicker, easier, simpler. That's the part of the stomach called cardia. Then there's body and the fundus and the pylorus. At the end of the stomach, there's pyloric sphincter, which is much bigger, much stronger, much more powerful. You're going to be able to very easily touch it when we dissect the pig. And it's a very, very tight control of the passage of food from the stomach into the intestine. Only a couple of milliliters at a time. So it opens, lets a couple of milliliters, closes, and waits a little while. And then once it gets into the duodenum, there's all sorts of things happening in the very first inch of the duodenum. That very first inch of the duodenum is called the ampulla of Vater, V-A-T-E-R. And at the bottom of that ampulla of Vater is the sphincter of Oddi, O-D-D-I. So that, that sphincter is kind of closing off, so the food, which is now liquid, it's coming from the stomach, is going to stay in the top inch of the duodenum for a little while, being neutralized and all that stuff, and then allowed to go to the rest of the duodenum. Duodenum got its name because it's about 12 inches long in an average human. After that, it goes into the jejunum and the ileum. Between ileum and cecum, there's ileocecal valve. It's not super good, but it does have a little bit of a control over how much food goes from the ileum to cecum. Cecum in us is really, really small, and then it goes into the colon and into the rectum. At the very end of the rectum, there are two sphincters, the internal anal sphincter and the external anal sphincter. Internal anal sphincter is made of smooth muscle, which means it is not under conscious control. The external anal sphincter is made of skeletal muscle, which means it is under conscious control. That's the one that you are squeezing and tightening while looking for the bathroom and running to it, right? When you really, really need to go. Which is exactly the same as is happening at the end of the at the bottom of the urinary bladder, entering the urethra. There are two sphincters there. Internal urinary sphincter is made of smooth muscle and it's not under conscious control. The external urinary sphincter is right underneath. It's made of skeletal muscle. It is under conscious control. Again, that's what you're squeezing and tightening while running to the bathroom so you don't pee in your pants. So it's equivalent. It's the same way in both of those systems. So the food is going through there. In order for the food to go through the mouth, from the mouth all the way to the anus and out, it needs to be pushed along. So there are different kinds of movements that are going to happen in the gastrointestinal tract. Stomach has 3 layers of muscles. Everything else has 2 layers of muscles. Those 2 layers of muscles are longitudinal and circular. Stomach also has oblique, so it has another layer of muscles which allow the stomach to really churn the food and turn it from a solid into a liquid. And then the liquid gets pushed by a couple of different types of movement. One is segmentation. So you have liquid chyme coming into a particular part of, let's say, jejunum, and you're going to have 2 parts of those circular muscles squeeze and make a segment, right? And so they have this liquid in this segment. They can't go back, can't go forward, and then you can slosh it back and forth, mixing the food with the enzymes and water so it can get absorbed there. And then you open it, and then you have peristaltic wave. Peristalsis is the movement always towards the next segment, right? It's always pushing to the other end, and it's kind of a wave of pushing the food back. So peristalsis is something that is going to be controlled by a lot of factors. Now you probably remember from the nervous system that the gastrointestinal tract, the digestive system, has a semi-autonomous nervous system of its own, right? You heard of that? That there's a whole bunch of nerve ganglia and bunch of nerves that are acting on the digestive system and barely gets any input from the central. So that nervous system is going to control the the peristalsis and the movement of the intestines. What is going to happen during stress? What is the action of norepinephrine on the GI tract?
One more time.
What is happening during stress? What is the effect of norepinephrine and epinephrine on the movement in the digestive tract?
It's inhibiting it.
It is inhibiting it, right? Because the digestive system is taking up a lot of water, it's taking up a lot of energy. You can stop for a minute, go run away from the lion, then you can continue digesting later. So it's going to be inhibited. The main neurotransmitter in this intestinal nervous system is serotonin. Serotonin is stimulatory to the movement. So all day long, you have a bunch of serotonin there, and it's going to act on the entire intestinal tract to increase movement. Where you have serotonin, what else do you get? What is the byproduct or breakdown product of serotonin?
Melatonin.
Melatonin, excellent. So during the day, a little bit of melatonin is going to be produced. There's going to be lots of serotonin. Some of it is going to be broken down into melatonin. So what you're going to have there is a very low dose of melatonin. At low dose, melatonin inhibits the movements of the GI tract. There's a lot of serotonin, a little bit of melatonin. They're kind of fighting it out. There's quite a lot of movement. During the night, most of that serotonin gets broken down into melatonin. At high doses of melatonin, which you experience at night, melatonin is stimulatory. It's more stimulatory than serotonin. You actually digest more and better during the night than during the day. So you can have a nice big dinner and then go to sleep and digest while you're asleep because melatonin works. Okay, another hormone that acts on the intestine is which hormone is going to increase the extraction of calcium out of food? Paired hormone, because it needs to increase the calcium levels in blood. So it's going to act there. All right. So there are a bunch of glands that are secreting all sorts of things into the food, right? You are eating mostly solid food, right? It's that mammoth again, right? And you need to dissolve all of that in water. And so salivary glands and liver and pancreas and intestine, they're all going to secrete a lot of water into this tube and mix it up with the food and with the enzymes. And once it's all digested and absorbed, then that water gets reabsorbed. Most of the water, 90% of the water, gets reabsorbed by the small intestine, not the large one. But the problem with the large intestine can lead to diarrhea because those additional 10% are not absorbed. So you start with a solid food, and what is happening in the mouth is that your tongue and your teeth and your salivary glands working together are going to break down the solid food into something that's a little bit less solid, something that is wet, right, and moist and round and smooth, something swallowable, which we call the bolus. And salivary gland, of course, is going to have some antibodies and some antibacterial stuff because you're taking dirty stuff from the outside. We didn't always have the ability to clean and wash our food or even know that it is important to do so. So we were ingesting bacteria and stuff with the food. So there's the first defense. The salivary glands, of which there are many little ones, but also three big ones. The three big ones are the parotid, which is kind of here. This is the one that gets enlarged when you have mumps. Sublingual, lots of little ones under the tongue, and submandibular, which is under the mandible, under the lower jaw. And those three big and a lot of little salivary glands are also going to contain some enzymes. they're going to contain salivary amylase. Salivary amylase, when it says amylase, means that it is digesting starch. And so when you eat starchy food like bread, at first bite, it's nice, but it's not sweet. But salivary amylase is going to start breaking down starch in the bread, and it's going to start tasting sweet. Saliva also contains a salivary lipase. When it says lipase, that means it's breaking down lipids. It is not starting any digestion yet in the mouth, but that lipase gets activated by the hydrochloric acid in the stomach, and it starts breaking down lipids in the stomach. Saliva is not a huge contributor of enzymes. It starts a couple of enzymes that are starting a little bit of digestion of two different types of molecules. It's not a big deal. If you don't have salivary glands, you can still digest quite okay. Stomach itself has an enzyme. That enzyme is called pepsin. Pepsin is secreted as pepsinogen, which is inactive form, and when hit by the hydrochloric acid, it gets activated into pepsin, and it starts breakdown of proteins. Again, you can live without it, but it starts breaking down proteins, big proteins into smaller proteins, peptides, and then delivering that into the intestine for further digestion. The stomach secretes hydrochloric acid. So pH inside of the stomach is somewhere between 1 and 2. Extremely corrosive, extremely dangerous. So stomach is constantly under attack by the hydrochloric acid. And it protects itself in 3 ways. The first one is it secretes mucus on the surface of its cells, and that mucus is going to contain buffers, like bicarbonate buffer, which is right there on the surface going to neutralize the acid, so it doesn't immediately get eaten by the hydrochloric acid. The second one are very tight junctions between the cells, so the acid cannot leak between the cells and destroy the layers, the muscular layers and serosa and whatnot outside. And the third is that the stem cells of the stomach are very actively dividing all the time and constantly replacing. These do get damaged, and so they're constantly replaced. There are situations in which, let's say because of stress or diet or something like that, you're not producing enough of these defenses and whatnot, or not enough hydrochloric acid, and a bacterium called Helicobacter pylori realizes it's very nice to living here and starts damaging the stomach. The result is what is called a peptic ulcer or stomach ulcer. For a long time it was thought that stress directly causes ulcers. The guy who discovered Helicobacter pylori had a really tough time persuading other scientists and doctors that there is a bacterium that does this. He said, no, it's stress. He says it's a bacterium, and I have the bacterium, and it causes peptic ulcers. And so because they did not believe him, he took a whole vial of Helicobacter pylori and swallowed it and gave himself a peptic ulcer. And then he treated it with the antibiotics, which also proved that it was a bacterium, right? Antibiotics worked. And he got a Nobel Prize for that. But that was quite, a personal sacrifice to show that he was right. He was really kind of, I guess, pissed off that they did not believe him. So Helicobacter pylori is going to act when the defenses of the stomach are weakened, right, and start making a hole in the lining of the stomach. So the stomach is going to take this very kind of solid bolus, and is going to liquefy it. It's going to churn it and digest it into a liquid. So the cells of the stomach are organized in little pits, gastric pits. They look sort of like this, although this is not the stomach. I'm sorry about the phone. But It looks like this bit, but this is intestine. This is a crypt of lubricant in the intestine. It looks kind of similar in the stomach, gastric pits. And gastric pits have two different types of cells. They have chief cells and parietal cells. Now, which produce what?
Chief cells produce pepsin.
That's correct. And
Parietal cells produce hydrochloric acid.
okay, and let's clarify, how do you know that? If the cell starts with C, it does not produce anything with C. It does not produce HCl. It produces the other thing. It produces the P thing. If the cell starts with P, parietal cell, it does not produce the P thing. It produces the C thing. It produces the hydrochloric acid. So you cross it over. P does not produce P, C doesn't produce C. It's the other way around. C produces P, P produces C. That's how you know forever, for the rest of your life, what makes what. What is the most important function of the stomach? A lot of people get bariatric surgeries, and what is the problem there?
Storage of food.
No. You can live without a stomach. A lot of people get bariatric surgeries and live without a stomach. What is the problem with a complete removal of the stomach?
No digestion.
You are perfectly fine. You can digest everything without a stomach, except one thing.
B12.
B12, exactly. That one thing is B12. So vitamins, A, D, E, and K are lipids, and they are going to be digested and absorbed like lipids. Others are peptides. They're going to be protected in some way, and they're going to be, absorbed as whole little vitamins. B12 is an enormous protein. It can't just, be absorbed through a pore or something like that. So it gets attached to a molecule called an intrinsic factor. Intrinsic factor is produced by the stomach. And it binds it and protects it from digestion and takes it all the way to the end of ileum, beginning of colon. And that's where the cells have machinery on the, molecules on the surfaces that are going to take this whole thing. That's pinocytosis or even phagocytosis, whatever you want to call it. You're engulfing the whole complex of the B12 and intrinsic factor and taking it in and putting it into capillaries, which at this place are fenestrated, and taking it into the organism. What is the name of the disorder of the lack of B12?
I should know this.
If you don't have enough B12, you get the disorder or disease called pernicious anemia.
I was not going to get that.
Pernicious anemia. So people who get the type of bariatric surgery where the entire stomach is removed, they need to get B12 or intrinsic factor additives. And sometimes it's B12 straight into the blood because without intrinsic factor they can't digest it. The B12 from the food is going to go right through. So that's the most important function of the stomach, the production of intrinsic factor, so you don't get the pernicious anemia. Without it, we can still digest. All right, so stomach liquefies, the bolus becomes chyme. So food becomes, that was solid to begin with, now is liquid, and it's going to be liquid for a while. The stomach is going to allow only a couple of milliliters at a time to go out of the pylorus via pyloric sphincter, which closes and opens. Why? Because the next part, the duodenum, does not have the defenses that the stomach has against the acid. So you don't want to dump a gallon of acid into the duodenum because it's like gone. So you take a couple of milliliters, 2, 3, 4 milliliters of the chyme and stick it into that ampulla of water in the first inch of the duodenum. And the first thing that happens there is neutralization. You neutralize the acid using buffers, mostly bicarbonate buffer. The bicarbonate buffer comes from 3 different sources. One is bile from the gallbladder, one is pancreatic juices coming from the pancreas, and one is Brunner's glands, which are in the wall of the duodenum itself. Those are little cells inside of the wall of the duodenum that are also secreting bicarbonate buffer. So there's a lot of bicarbonate buffer right there in the very first inch of the duodenum, busily and speedily neutralizing that acid. So you're moving from pH of 1 to 2 to a pH of about 7.5, 8, a little alkaline. And it remains alkaline to the end of the digestive system. So it's a very, ampulla of water is probably the most interesting and most important part at 1 inch of the whole digestive tract because so much is happening there. So you have 3 sources of buffers that are neutralizing the acid. And you have pancreas coming in, and you have gallbladder coming in. So all sorts of things are coming in there that are really essential. So enterocytes, which are the cells of the intestine, they can make their own enzymes, right? Digestive enzymes. But most of the digestion is done by the enzymes that come from the pancreas. You've seen the slides of pancreas, and you had to kind of look around to find the tiny little islets of Langerhans, the endocrine part. Most of the rest of it is exocrine part. Those are the cells that are making digestive enzymes, putting them in little tubes. Those tubes fuse to each other. You end up with the pancreatic duct that goes into the duodenum, into the ampulla of water. And it secretes all of those enzymes, various lipases and whatever, amylases and nucleases, and you need to know what name means as far as what kind of a substrate they break down. This also includes several proteases. Proteases are enzymes that break down proteins. The most important of this is trypsin, and gets secreted by the pancreas in the form of trypsinogen. Whenever there's a gen, you know that's an inactive form. And then once it hits the ampulla of water, there's still hydrochloric acid there. There's going to be activation into trypsin. Trypsin then takes the other proteases like chymotrypsin and I don't know, a couple of others, and activates those too. So all of the proteases are coming out of pancreas inactive and then activated in the ampulla of water, and now they can actively break down proteins for the rest of the intestine. The reason for this is that all of these others can be active in the pancreas, no problem. But if the proteases are active in the pancreas itself, they would digest the pancreas. So they have to be secreted in an inactive form and only activated inside of the intestine. All right. Liver. Liver is a very metabolically active organ. It's taking up, all sorts of things from various places. It's taking all the nutrients from the intestine coming through the hepatic portal vein. And it's taking toxins. It's taking and detoxifying them. It's taking breakdown products of hemoglobin, which is bilirubin, which is toxic. And, it's manipulating those things, it's breaking them down, it's trying to reuse which can be reused, but not everything can be reused. Something that has to be just gotten rid of. It's toxic, it's not good for you, or there's no use for it, and you want to get rid of it. And the liver is going to get rid of it by putting it all together into a liquid called bile. Now, bile is going to partially go out of the left and right lobe of the liver through the left and right hepatic duct. And then those two ducts are fusing into the common hepatic duct, which then flows into the common bile duct, and common bile duct goes into the ampulla of water of duodenum. So, as bile is produced, there's going to be a little drip, 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 drip of bile going into the intestine at all times. And then a lot of the bile is going to be diverted, and instead of going into the duodenum, is going to be filling the gallbladder. the gallbladder is going to be filled with bile. And it's just going to sit there. And when you eat your mammoth, this is the gallbladder, and this is the gallbladder. Gallbladder is full of bile, and it has a duct too that is called the cystic duct, because bladder means cyst, right? Cystic duct also flows into the cystic duct and common hepatic duct. When they fuse, they form the common bile duct that goes into the duodenum. And it just sits there. But let's say you have eaten your fatty meal, your mammoth, which is fatty, and your stomach has started churning it, and the first kind of bits of chyme are going into the duodenum, and the duodenum senses there's a lot of fat in there, and it's going to secrete a hormone called cholecystokinin. Chole, bile, cyst, bladder, kinin, movement. So it's going to stimulate the movement of bile out of the gallbladder. So it's going to tell the smooth muscles in the walls of the gallbladder to contract and push all of that bile out the cystic duct into the common bile duct into the duodenum. Why? Well, a lot of the breakdown products of various metabolic processes are salt. Remember bulbourethral gland or Cowper's gland? What do salts do? They act as soaps and detergents. All right. So why do we use salts and detergents in the kitchen?
Clean.
What exactly? Well, yes, we're cleaning. Remember that water and oil don't mix. I used to bring a lava lamp, and I don't know where it is anymore, because lava lamp is built on that principle, right? You have a bunch of oil in water, it's going to separate, and you have these big globules of fat slowly flowing up, right? So that's what happens when you have a lot of water in the digestive system and you have fat in the food. It's going to form big globules. Big globules have relatively small surface and a lot of volume. Very, very valuable energy, and poor lipases are trying to hit that, and they can't, because there's very little surface and a lot of volume. So you need those salts and detergents. What do salts and detergents do? They break down the big globules in tiny little droplets. Now you have much more surface, much less volume per droplet, right? Which means that now lipases can act on the lipids and start digesting them. This is a slow process. Eating a bar of chocolate is, or something candy is just carbohydrates. It goes really fast. Eating protein takes a little longer. Eating a fatty meal or a fat-rich meal takes the longest. Longest in the stomach, longest in the entire process. It takes a while. And this is one of the reasons, because you need the bile to break down those droplets in order for the
Which means if you eat fatty food, you will not feel hungry. It's a long time you will feel hungry.
You feel full for a long time.
You feel full for a long time after a
that-
fatty meal.
yeah,
Yes,
yeah.
you go have a steak and something like that, and you're not going to be hungry for a long time. That's exactly right. And it's going to be hormones like gastrin and cholecystokinin, and there's a few others, and leptin that are controlling appetite and hunger, right? For instance, gastrin, when the food starts falling into the stomach and starts stretching the stomach secretes gastrin, which then increases the movement of the stomach and the intestines, getting it ready. Like food is coming, let's start churning, let's start moving. So it's a whole bunch of hormones are going to act on that. I'm not going to ask about all those little details. Anyway, which kinds of animals have to have a gallbladder?
Carnivores.
Carnivores, right? Because a lion is going to keep hunting for a day or two and not being lucky, and then finally catches the zebra, and zebra is going to have a lot of fat, and the lion is going to eat a whole zebra in one sitting before the hyenas come, right? And so it's going to gorge itself with a lot of fat, because it's not going to eat just steak. It's going to eat all the innards and all the stuff, all the mesentery, which is full of fat, which is like the best because it's a very energy-rich material, right? We usually use carbohydrate, but fat provides more energy when you need it. And so it has unpredictable times when it's going to be a meal. Meals are happening only occasionally, and meals are happening when they happen, they're big and they're very fatty. So carnivores, like cats and dogs and stuff like that, have to have gallbladders. We are more of omnivores. Our ancestors were much more carnivorous than we are, but we have gallbladders because we do eat distinct meals, and we eat fatty meals. We do eat a lot of animals. Which animals don't have to have gallbladders?
Herbivores.
Horses, herbivores. So, horse is going to graze 24/7, occasionally taking a nap here and there, moving a little bit, but mostly grazing. It's grass and hay. There's no fat in there. Essentially no fat. There's a little bit. Membranes are made of fat, right? Okay, it's going to be a little bit of fat. Maybe it's a beetle and gets fat like that, right? So, and it's eating continuously. There's not a distinct meal. It's constantly eating. And so what happens if you have a gallbladder and you don't need it? If you have a gallbladder, you have an increased risk of gallstones, which can kill you, right? So if you don't need a gallbladder, you better not have one. So herbivores have evolved to lose gallbladder. And so all of the bile that's made by the liver is going from the hepatic duct into the common bile duct into the duodenum at a slow rate of drip, drip, drip. That's enough to digest whatever little fat can come in with grass. So they're perfectly fine without the gallbladder. They don't have it. Okay, so now we are going through the intestine, and digestion is happening. All of those various big molecules are broken down into smaller and smaller molecules. A lot of the times, the pancreatic enzymes, which are doing the bulk of digestion, are not going to break it down all the way. So for instance, the pancreatic enzymes are going to break down polysaccharides into disaccharides, with two sugar molecules bound. And then the disaccharide is going to attach itself to the surface of the enterocyte, which is going to have its own enzyme, which is going to break those two molecules apart. So instead of a disaccharide, you have two molecules of monosaccharides that can be absorbed.
Is that an interior site or is that a microvilli?
It's on the outside. It's a microvilli. Yes, it's on the outside. They put those enzymes on the surface. So monosaccharides are going to actually be absorbed into the cells, into the enterocytes. And so monosaccharides, amino acids, phosphates, ribose, deoxyribose, the purines and pyrimidines, all of that stuff is going to be absorbed through the through these cells. Those are pretty much cuboidal cells. They're going to go through them in a controlled manner, but pretty quickly, and they're going to go into these capillaries, which, remember, are fenestrated capillaries. So those kinds of molecules can pass through the fenestra and get into the bloodstream. All of that blood is going into the hepatic portal vein and goes to the liver for the first kind of check if everything is okay for metabolism. The liver decides what to do with all of those crazy molecules and starts rebuilding them. Those are building blocks. Those are little Lego pieces, and now it needs to build, the helicopter and the boat and an airplane that are human and not cow or tofu or mammoth or whatever you have eaten. So liver is going to control all of that, and then it's going to send all the good stuff into the hepatic vein, into the inferior vena cava, and into the rest of the circulation, all nice and fine. And if it's bad, it's going to break it down and put it into the bile, which is then going to go out with feces. Bilirubin is the breakdown product of hemoglobin. So when red blood cells get broken down in the spleen, that bilirubin gets transported to the liver, and the liver is going to put it into the bile because bilirubin is toxic. Bilirubin has color. Bilirubin is what gives the bile the green color, which is why the gallbladder looks green. And when we open up a pig, you're going to see the gallbladder is green. And then as it goes through the whole process of digestion, bilirubin is what gives the feces the brown color. So let's say you have gallbladder removed, right? Which happens to a lot of people, right? If you have a gallbladder removed, and at first, if you're eating anything fatty, what is your feces going to look like? It's going to be white. Because it takes some time for the whole thing to kind of balance. And after you have removed the gallbladder, you shouldn't really eat very fatty stuff anyway. So there's no color, there's no pigment to make the feces brown. It's white, yellowish white. Okay, so all of that food is then going through the stomachs or the intestines, and all of that gets absorbed. What about the fats? Now, fats are complicated. So let's say you have triglycerides. So you have glycerol and you have the 3 fatty acids. And they get taken in by the enterocytes there on the surface. And those enterocytes reconstitute the fats. They make our own fats. They take their glycerol and they modify the fatty acids, and they make new triglycerides, the human type, your type. Those molecules are big. Those molecules are not going to go through the fenestra of the capillaries. Instead, they need to go into the lymphatic vessels. Lymphatic vessels here in the intestine are called lacteals. And those are, as you know, what kind of capillaries? Sinusoid capillaries. There's whole big chunks of wall missing and whatnot, which means big fatty molecules like, triglycerides can go through those holes and get into the lymph. And it's going to be taken through, lymphatic system, through the lymph nodes and whatnot, end up in the subclavian veins, and that's how it enters the cardiovascular system. It bypasses the liver, which is a great way to give medicines without the liver breaking it down, right? You make fatty medicines, so they have to go by lacteals directly into the cardiovascular system and not through the liver first, so they don't get broken down by liver. So that's also a reason why it's slower than to digest a fatty meal. So about 90% of the liquid gets reabsorbed in the small intestine, and then the large intestine is going to do the rest. Large intestine is going to do a little bit more of absorption of some things, and it's going to have microflora, a bunch of bacteria, archaea, yeasts, and all sorts of other things, protists that are in your intestine that are defending you from the bad bacteria, and they're also producing some vitamins, and they're helping with the digestion and absorption of some vitamins from food. So at the beginning of the colon, you still have some of that kind of stuff happening. And then the rest is just making the feces and going out. The colon only, the large intestine only has kind of strips of kind of fibrous connective tissue that are called teniae and haustre. Again, teniae are longitudinal and haustre are circular. And those are associated with some smooth muscles and whatnot. And they are making the shape of the feces. You go out into the woods and you are going to see feces of different animals, and you're going to recognize them. This is a rabbit, this is deer, this is bear, this is dog, this is horse, this is cow. Every one of them has different shape and sizes. What is the shape of the feces of a wombat? Wombat is a marsupial from Australia. It's a square, it's a cube. So wombat is going to dig a cave underneath a rock, and it's going to be highly territorial. This is my rock, and it's going to poop a cube on top. Now, if it was round, it would roll over, right? But if it's a cube, it sits nicely on top and tells everybody, this is my rock. So that way the male wombats can stake their territories by using cubical poop. And over the last few years, a group of scientists in Australia has been studying how do they make cubes. It has nothing to do with the anus. It has to do with the Taenia anhausta and with the cells of the colon and of the rectum. And in the additional readings, there are several links to a couple of those studies of how they figured it out, which is, we can use to figure out why our poop is like the shape it is. Not very exciting. We don't do cubes or anything fancy like that. Okay, and yeah.
This is Taenia and this is Hostra?
Yes.
Okay.
Okay, so what else is happening here? So the fermentation vat, the fermentation area where we have all these microorganisms helping us digest, are at the very end, in the colon. In other animals, is in different places. In perissodactyls, remember those? Those are odd-toed ungulates. So those are animals that have an odd number of toes. Those are horses, donkeys, zebras, tapirs, and rhinos. They have a cecum that's about this long, about 1 meter long, enormous, and that is full of bacteria. That is where most of the fermentation goes. This is where those microbes digest cellulose for them. And that's where a lot of that broken down cellulose, which is glucose, actually gets absorbed for these animals. What about artiodactyls? Especially ruminants. Those are the even-toed ungulates. Those are cows and sheep and camels and giraffes. What do they have? They have a stomach like us, right? But that's only the fourth of the 4 chambers. They have a 4-chambered stomach. Only the fourth is the equivalent of ours with hydrochloric acid and pepsin and whatnot. The first 3 are fermentation chambers with different types of microflora in them. And they're absolutely fantastic because the cow swallows moldy hay and doesn't matter because they're immediately bacteria and funguses and proteins that are breaking down that mold, and the cow is fine, which is why cow hay is cheap and horse hay is expensive because it has to be immaculately clean. It cannot have any fungus in there or any rot because horse is going to die because they're going to get intoxicated with toxins before it gets to the cecum. And we are even worse, we're in the colon. So much more sensitive to outside toxins than the cows and the sheep and the goats. Goats can eat anything, right? Okay, so we do eat toxic things, and so they go, they get digested, they get absorbed, and they go to the liver, and liver detoxicate. So detoxifies toxins. It breaks them down, turns it into some metabolites that then can be excreted with bile out. So for each one of those toxins, the liver cells are going to have a specific enzyme that detoxifies it, like it breaks it down. So different kinds of toxins are built differently. You have to have a specific enzyme to start breaking it down so it doesn't kill you. So one of the toxins that we often take in is ethanol, right? We are that way. A lot of other animals also, eat fermented fruit and get drunk like elephants do and whatnot. But, we are really sophisticated alcohol drinkers. And so we take in the alcohol. And what happens is that alcohol gets absorbed and goes to the liver, and liver breaks it down. It uses the enzyme called alcohol dehydrogenase. Very easy to remember, breaks down alcohol. And that way, the alcohol does not get into the circulation, it does not hit your brain. So you need to drink a few more drinks in order for all of that quantity of alcohol to overwhelm the capacity of the liver to break it down, and starts leaking into the circulation and passes through the blood-brain barrier, and you start feeling it, right? So you go out with your friends and you don't drink half a beer, you drink 3 beers, 4 beers, right? And then like, yeah, now I'm feeling good. Now I can dance. And now I'm over my shyness about dancing. I am dancing now because I had 4 beers, right? What do alcoholics do?
Drink in the morning.
They drink in the morning. They drink in the morning because they need an instant hit in the brain. And they don't want to drink 5 million beers. They're going to probably get a shot of vodka or something like that first thing in the morning. Why? Because, like everything else, alcohol dehydrogenase is not produced, meaning transcribed and translated by the liver cells all the time. There's a circadian clock in the liver, which is going to turn on and off various genes at different times of day. So if you need to, digest carbohydrates, those enzymes and all that stuff for breakdown and for building carbohydrates, that's going to happen in the morning when you're having your cereal. And in the evening, when you finally manage to hunt down and roast that mammoth, that's when you're going to have enzymes that are dealing with the proteins and fats. And also that's the time when you're dealing with the alcohol, because the whole village is celebrating a successful hunt and is making mead, right? Which is an alcoholic drink, so everybody's getting a little drunk there, right? And so alcohol dehydrogenase is mostly produced by the liver between noon and midnight, which is why we socially drink in the evening, right? Because we can take it, right? Alcoholics cannot do that. They have to drink at the times when alcohol dehydrogenase is not transcribed and translated, because they want that shot of vodka to go straight to the brain, because they're actually addicted. They need dopamine, right? In their brains, they have cravings because they're not just social drinkers, they're actually sick. They are addicted to alcohol, and so they have to drink in the morning. So it's a fantastic bonus question, this whole detoxification and circadian clock in liver and alcohol. It's a great bonus question. I think, did I say everything? I think I said everything I wanted to say. Any questions?
Can